Wonderful. Thank you very much, Becca and Monica, for that introduction. That was really great. Thank you. And thank you, Rachel, for inviting me to speak today. It's a real pleasure. Yes, so much of what I would introduce myself has already been said. But yes, I am a postdoctoral researcher at ETH Zurich. I will preface just now that I am Australian. I appreciate at times I am hard to understand, so I will try to speak slowly because we have a tendency to speak fast. If you do not understand anything I've said, please feel free to interrupt. I will not be offended at all. I get it on a daily basis in Switzerland too, so I'm still learning. But as Monica, oh, and also if you just want to interrupt with a question, please feel free to do so. I would love that, in fact. So as was introduced, I did just want to retouch on these things that, yes, I used to work on planes, trains, and boats. This is my actual modeling shot, the best shot of underwater, and you can see it's really not that glamorous. But it is easy to look good when you have most of your face covered, I've found. All of those, the reason I want to highlight those things is that all of those experiences that I'd had in the past have very much led me to my opportunities that I had in my PhD, which, as we also touched on, looking at blue whales and Antarctic krill down on the Antarctic ice edge. And now for me at ETH Zurich, getting to work on the Tara Oceans boat, honestly, it has been a dream come true. I visited this vessel when it was in Sydney Harbour in 2018. So all of that would not have been possible, I don't think, if it were not for my engineering and diving background that I've had too. So please, just as a message, never overlook the things you've done outside of science in terms of your career too. It's really quite important. So Rachel has invited me today to talk about ocean sulfur cycling. So I did hear you've touched on DMSP as part of your reading, which is excellent. I will go over a general ocean sulfur cycle and hopefully impress upon you its importance. And then I'll also just talk about some of the research that I did with UTS as part of my PhD. So just quickly looking at sulfur cycling by marine phytoplankton and bacteria. So focusing on the actual microbial ecology that underpins the cycles of both DMSP and one of its breakdown products, DMS. And then at the end, I will touch again briefly on TREC, which is traversing European coastlines. So if any of you are unfamiliar, at the moment, there is a big multinational, multi-university and institution collaboration going on, which is looking at land and sea all across the European coastline. So we've started in late February this year, and we have just concluded now where we have managed to go across most of the Northern European coastline, and next year we will visit the Mediterranean coastline. But in particular, my role is actually looking at bioaerosol microbiomes across Europe. So looking at airborne microbes. And then, of course, we'll have some time for questions. So please feel free to put them in the chat, but I also encourage you to interrupt me. That's great too. So in terms of ocean biogeochemical cycles, quite often when you look at ocean biogeochemical cycles, you'll often hear about other elements such as carbon, nitrogen, oxygen, phosphorus, and silicate. When you look at these cycles, of course, they are all very, very important, which is why they have been focused upon. But what is commonly overlooked, or what hasn't received as much research attention, is the sulfur cycle. So sulfate is abundant in the ocean. Sulfur is the 5th most abundant element in the universe. It is the 15th most abundant element in the Earth's crust, and it is essential for life. But for some reason, it has not received as much research attention as the other elements, and particularly carbon and nitrogen, I would say. If you are to look at some biogeochemical cycles. So what I chose for this image was a obvious carbon pump cycling that's happening here, but you can see many of the other elements, such as the benthic flux of other biogeochemicals, as well as some nitrogen fixation happening too. And all around, if you were searching for sulfur in this picture, there is one area which has received the most attention of the ocean sulfur cycle, or sulfur cycling in general. And that is dimethyl sulfide, okay, or DMS. So you have mentioned DMSP as a element, but dimethyl sulfide was the, I guess, what put ocean sulfur cycling on the map, I should say. And this is very much at first, traditionally and historically, we thought that hydrogen sulfide was the major vehicle of sulfur from the ocean to the atmosphere. So I should state, sorry, dimethyl sulfide is a volatile. It is a gas that is emitted from the ocean. So in 1972, so this is James Lovelock, who was a climate change scientist who I believe passed away either last year or the year before at age 103. He suggested that DMS is the natural sulfur compound which fills the role originally assigned to hydrogen sulfide. So what it is, it's the major vehicle that transports natural sulfur from the ocean to the atmosphere. Now I can promise all of you that you've interacted with DMS in some form or shape, okay? It is that tangy, sulfury smell that you get as you approach the ocean. So if you have ever been to the beach and you could smell the beach before you could see it, and then you turned up and saw something like this, I can guarantee you that very tangy smell was dimethyl sulfide. So it is quite commonly called the smell of the sea. DMS, as well as being the largest efflux of sulfur from ocean to atmosphere, it also gained quite a lot of popularity through a 1987 paper, which was written by Charleston, Lovelock, Andrea, and Warren, who put forward what is commonly referred to as the claw hypothesis. So the idea was that phytoplankton produce DMS, and this DMS is able to ventilate into the atmosphere where it will form cloud condensing nuclei. So that cloud, the cloud condensing nuclei promote cloud cover, which then in turn reflects solar radiation. So this is what's known as a negative feedback. So it is actually thought to cool the climate. So this became a very, very attractive thing for both climate change scientists, and I believe eventually led to James Lovelock hypothesizing a Gaia hypothesis. But what it has, and so this actual theory of it, although globally we are still not convinced, you can see within some regions it has actually been documented. So a group from Southern Cross University in Australia has gone to the Great Barrier Reef and looked at the Capricorn Bunker here. So this is Hilton Swan from Southern Cross University, and they've actually measured DMS and cloud condensing nuclei and also been able to map the subsequent clouds that happen on that day. As I said, globally, we are still uncertain as to whether the claw hypothesis holds. And so as you can see, that excitement in DMS that was produced by the claw hypothesis has resulted to 87,000, well, over 87,000 public measurements that you could access now. And one thing I will highlight about this particular cycle is that the concentrations that you see in areas such as the Southern Ocean or the North Atlantic, they are much higher concentrations. So typically both DMS and DMSP, which is its other product, is its precursor, are found in greater concentrations towards high latitudes. And you can also see that they are seasonal. So they vary over both space and time. So as you can see, as I just showed you, DMS has received quite a lot of attention. Part of the reason that perhaps the claw hypothesis does not hold, though, is due to The ocean does not just produce DMS, it is derived from a compound called dimethyl sulfoneopropionate, or DMSP, okay, it's a zwitterion, which is produced, there's over 1 billion tons per year of it produced in marine surface waters. A large amount of it is actually produced by marine phytoplankton, okay? So this claw hypothesis, which looked at phytoplankton and DMS, there is a very important step in between there. And it does seem that DMSP is quite an important compound for marine phytoplankton. So they can dedicate up to 10% of photosynthesis into making this compound. You can see not only is it a sulfur compound, it is rich in carbon too. And it can comprise up to 50% to 100% of the sulfur that can be found within a phytoplankton, so of cellular sulfur. Another reason that this interaction, the claw hypothesis may not be so straightforward is because it actually has been hypothesized to play a number of intracellular roles. None of them are growth. So this is a secondary metabolite for phytoplankton. So among these intracellular roles, you're looking at being a cryoprotectant, an antioxidant, so that is scavenging reactive oxygen species, and then also an osmolyte. So when salinity changes, phytoplankton are seen to change the concentration of sulfur within them too. So what actually ends up occurring of the DMSP that is produced by marine phytoplankton, it's been estimated that just 10% is made into DMS that can then be ventilated into the atmosphere. Sorry, did someone say something then, or? No, that's okay. So with DMSP rather, it is produced within the phytoplankton cell, but hopefully you have been introduced to the marine food web and you do know that much of the dissolved organic matter that they produce is lost to the water column. So a ubiquitous form of dissolved organic matter is dissolved DMSP, which is in the water, particularly in marine surface waters. And it attracts a lot of heterotrophic bacteria, okay? So in contrast to the phytoplankton, heterotrophic bacteria are known to use DMSP for growth, and some major clades are shown to do it. And it can represent 15 to 90% of sulfur for many marine bacteria. It's been shown to be a potent chemoattractant. So motile bacteria who are chemotactic are actually been seen to swim, been evidenced to swim towards DMSP to locate resource patches of food. And then also it's been shown to mediate the nature of phytoplankton bacteria interactions. So recently it's been shown that a bacteria called a sulfitobacter, which is part of the marine Rosiobacter clade, has been shown to shift from being coexistent to pathogenic, dependent on whether it is receiving DMSP with other phytoplankton-derived molecules, compared to if it just receives DMSP, it will then become pathogenic and will kill its host phytoplankton that it is coexisting with. Okay, so it can mediate those interactions between phytoplankton and bacteria. Bacteria's role, though, is also transformation of DMSP. So this link between DMSP and DMS, we know, is through what is called DMSP cleavage. And bacteria are also able to perform this function. However, as I said, only 10% of DMSP is funneled through this particular pathway. Much of it is actually channeled through what is called the demethylation pathway. Okay, so DMSP demethylation reduces DMSP to other forms of sulfur, which are easily accessed and assimilated as food, for example. Not only though, so this is right in the middle of my PhD, we learned this, that bacteria are also able to produce DMSP too. So that changed a particular paradigm where we thought they were just interacting through this chemical. All of a sudden, we knew that bacteria were able to produce it, so things got more complicated still. Beyond that, we then also learned that both bacteria and phytoplankton can produce a chemical called dimethyl sulfoxonium propionate, which is just an oxidation of DMSP. And then furthermore, bacteria can oxidize DMS to dimethyl sulfoxide too. All of these different pathways ultimately can lead to that reduction in DMS that we see from all of that DMSP that's produced, okay? So I do not want to overwhelm you with this slide. I just want to impress upon you that this is still very much an emerging field of science. It is very exciting still, and it's seemingly getting more and more complicated the more we learn. So on top of knowing about those pathways, we're also identifying genes of these degradation pathways too, okay? So we are able to go out and measure both the abundance of these genes, and if we look at RNA, we can look at the activity, so the actual expression of these genes too. So many of these genes, so this is a trimethylamine monooxygenase, DSYB, which is a bacterial production. And then we also know of a cleavage gene, well, actually many cleavage genes, I'm sorry, but I'm generalizing. And then also a demethylation gene called DMDA. Now today, in a moment, I will mostly focus just on these two pathways for my subsequent case study, okay? But please, if you are interested, there is a lab run by Jonathan Todd at University of East Anglia in England, and they are largely responsible for the identification and the clarification or validation of all of these genes. They've done amazing work to identify this. I'm simply a benefactor who goes out and measures them in the environment. So one thing we do know about DMSP cycles, obviously, as I've shown you, they vary in both space and time. And this is very true, even off the coast of Sydney. So this is home for me. And this particular point is called Port Hacking, New South Wales, which is just 8 kilometres off of Sydney. If you have ever seen the film Finding Nemo, you're probably familiar with the East Australian Current. But what that is it's a poleward flowing current, which will pump hot water down along the Australian coastline. So we have seasonal intrusions of these waters each year. So when looking at dimethylated sulfur, I looked at DMSP at this particular area. So I went back every month to sample the water. And I did actually find that in the springtime, we do tend to have seasonal peaks of DMSP. But that aside, I really, really wanted to know what was the underlying ecology. So who is there? Who is potentially responsible? And what genes might they actually have that might be supporting these trends that I see in my biogeochemical data? So in order to do that, I went to Port Hacking at between 2017 and 2019. And all I'm showing you here just is that on top of my research, the research that's going on at Port Hacking is actually been going on for years. It is one of the longest time series in Australia. And since 2012, there have been molecular data sets being generated from there, looking at phytoplankton and bacterial communities since then. But each month we would go out and using a rosette sample the seawater. So I would particularly just look at the surface seawater because that is the interface between ocean and atmosphere, obviously. And then I would use gas chromatography to then quantify both DMS and DMSP. On top of that, I was interested in the microbial community. So in that exact same source of water, I would extract the DNA from the microbes. And then using sequencing, so DNA sequencing, I would have a look at the composition of the bacterial community, looking at the 18S rRNA region, and then also the 16S rRNA region for bacteria. And then using quantitative PCR, I had a look at the abundance of many of the degradation genes that they possess. So essentially what I found throughout this time series was that, yes, DMSP did increase. However, if I bring your attention to these grey bars, they did not always correspond with phytoplankton blooms. So it's often thought that phytoplankton are the main producers of DMSP. When you have a phytoplankton bloom, it should be associated with increases in DMSP concentration. There was a positive relationship between DMSP and phytoplankton. And this in particular was shown in October 2017 at Port Hacking. So you can see both a phytoplankton bloom and high DMSP concentrations. However, during 2018, we actually saw a decoupling of the two things. So chlorophyll A, which is the proxy for phytoplankton biomass, decoupled from DMSP. So it was a very large phytoplankton bloom and a very small amount of DMSP comparative to a few months later in November, where you can see a much larger proportion of DMSP compared to that phytoplankton bloom. Okay, so this was an indication to us that, okay, yes, we have seasonal blooms, but there is something else happening here. In order to investigate that further, I had a look at the actual fractions of DMSP. So you can have DMSP within the cell, which is your particulate matter, and then also your dissolved matter. And interestingly, you could very much see that bloom in October 2017, far more of the DMSP was associated inside of the phytoplankton cell compared to the following year's peak, which I saw almost as much of it was in the dissolved fraction. Okay, so that for me meant that, oh, okay, I am probably going to see very different community composition, maybe not in my phytoplankton community, but in my bacterial community that coexist with the phytoplankton. So the first thing I did want to elucidate was, does my phytoplankton community actually explain that decoupling? So using that 18S sequencing, and I'm sorry if this is too rudimentary for you, but using DNA sequencing, you can identify different 18S sequences. For instance, identifying things like the diatom, Thalassiosaura, compared to, say, a dinoflagellate like Alexandrium. Okay. At the time, we had decided to reach out to some collaborators, so Erin McParland and Professor Naomi Levine from University of Southern California. They had just put together a bioinformatic pipeline which looked at what we know from culture. So what is a high DMSP producing phytoplankton, and what is a low DMSP producing phytoplankton? And they were linking them to sequence information. So through similarity, you could go and look at the sequence similarity and say, well, okay, this is likely a low DMSP producing Thalassiosaura versus a high DMSP producing Alexandrium. So with that context, looking at things within that frame of view, we started to have a look at the phytoplankton community composition. Okay, so I am actually showing you the complete time series and all of the 18S sequences in major groups of phytoplankton. Okay, and using the pipeline that was designed by Erin and Naomi, we actually managed to identify high DMSP producing candidates and low DMSP producing candidates. And in focusing on that, what we actually found was that there was a significant positive relationship between high DMSP producers and DMSP. So that was, I guess, quite obvious and unsurprising to us. What was surprising, what we also found though, was that despite a positive correlation with chlorophyll A, things such as your diatoms were really not associated with DMSP in the marine surface waters. Furthermore, because we were faced with the new challenge that bacteria also produce DMSP, we used quantitative polymerase chain reactions to quantify the DMSP production gene from bacteria. And what we actually found too, that there was a significant correlation between that bacterial production gene and the amount of DMSP in the water. Okay, so this for us was exciting because it was the first evidence in marine surface waters of that association between bacteria and concentrations of DMSP. So we'd pretty much concluded that high DMSP producing phytoplankton lineages and the bacterial DMSP production gene were associated with high DMSP concentrations with that study. But we were still very curious about the heterotrophic bacteria, the degradation of DMSP too. So we had a look at the bacterial community composition too. So as you can see here, I'm actually showing you both DMS and DMSP concentrations on the left axes. And then on the right, I'm showing you the relative abundance of the top 21 orders of bacteria. Okay, so you can see how some of them do shift over time also. In looking at things across the entire data set, what we had actually managed to see is that one group called the Flavobacterialis were positively correlated with chlorophyll A. Meanwhile, when focusing on dissolved DMSP, so that available source of DMSP, we actually found that this group called Rhodobacterialis were positively correlated with DMSP, or dissolved DMSP. This was wonderful for us because it actually showed something which was well documented before, which is particularly that these two groups, so the Flavobacteria and Rhosiobacter groups, Rhosiobacters are a subclade of Rhodobacterialis. They've been referred to as master recyclers, and they are phytoplankton-associated bacteria. And this is from this exact same article. One of the major links they think between Rhosiobacters and phytoplankton is indeed DMSP. So we were able to actually show this across our time series that yes, indeed, our Rhodobacterialis were linked with DMSP throughout our time series. And notably, sorry, I will just go back. Notably, their peak was found during the highest peak of dissolved DMSP that we saw in our time series. So that somewhat showed me where I'd earlier speculated to you that the difference between particulate and dissolved would mean differences in the microbial ecology. This was real evidence that yes, that increased dissolved DMSP really might support an increase in Rhodobacterialis. And also beyond that, sorry, you can see here that not only are many Rhosiobacters able to grow on DMSP, they are known to degrade DMSP through different pathways too. So both the cleavage pathway, which I'd shown you before, and the demethylation pathway. In having a look at actual concentrations of the DMDA, so this is the actual Rhosiobacter-associated demethylation gene, we found that it was significantly correlated with the Rhosiobacters too. And sorry, this is the actual, as you can see. Across the time series, however, DMDA was not actually linked to, so the abundance of the gene was not actually linked to DMSP dissolved. So to explain this is likely because of the activity of the gene. So just because the gene is there, it doesn't always mean that it's used. So that variability was actually seen in our data set. But one thing I will come back to again is that November time point in 2018, that we were seeing a very high abundance of that, corresponding with a high abundance of Rhodobacterialis, corresponding with high DMSP dissolved and very low DMS. So this is an instance where we actually think if we had been able to sample for RNA that day, we would have seen high activity of that gene, reducing the amount of DMS that could potentially get to the atmosphere. So I hope a takeaway from this that I can present to you is that really the ecology matters, okay? If I was to highlight peaks of DMSP in 2017, typically what you end up seeing is relatively high DMS, very high DMSP, most of it being particulate, and also high DMSP producing lineages of phytoplankton associated with Flavobacterialis. So flavobacteria are actually known to break down high molecular compounds, and they don't preferentially feed on DMSP. So it could be that they are receiving other compounds from the phytoplankton at the time. Alternatively, in September 2017, we had diatoms, a big diatom bloom, very low concentrations of both DMS and DMSP. So this phytoplankton bloom is not contributing to ocean sulfur cycling at that point, or sorry, DMSP cycling, I should say, because there are other compounds that they do. However, then in November, as I've highlighted also, with lots of dissolved DMSP, we were seeing lots of DMSP degradation by marine rosiobacters. And it was dominated by high DMSP producing phytoplankton and the Rhodobacterales clades. So it is this interplay between ecology being mediated by DMSP, but also them having control of the breakdown of DMSP derived products. Okay. And then now I've seen I've gone for 35 minutes, I'm sorry. I will quickly show you what I'm currently up to. So this is, I'm presenting right now Trek and Tara Europa so far, okay? So my role is understanding the dispersal of airborne microbes across European coastlines. So I'm unsure if you've seen this before. I'm delighted if I'm the first person to show you this huge effort, but this is the Trek and Tara, I guess, map that we have been going from. So the voyage commenced in Roscoff this year. Well, actually, it started in Lorient this year. And then we've had both the Tara vessel and also a land team from EMBL in Heidelberg in Germany, basically following one another around the entire coastline of Europe. So this year, they've been going all the way through, as I said, around Northern Europe, and now they've actually stopped in Lyon. So they're somewhere here now. But what they have been doing is basically more than 120 land-sea transects. I can tell you that Tara at the moment has actually done 100 different sites. At selected sites, we have super sites which are highlighted here. So Roscoff, Tallinn, Kristineberg in Sweden, Bilbao and Porto. And then next year, some of these have changed, but it will be Barcelona, Naples, and Athens next year. We're actually stopping in those destinations, and we're sampling for 5 days straight, both from land and sea. Essentially, we're looking at soil, seawater, and aerosol microbiomes, and we're trying to understand the flow of different genes and microbes from one environment to the other. My role is the aerosol microbiome. So in particular, I'm interested in the atmospheric boundary layer. So that's the layer of air which is just above the ocean. As I said, Tara is collecting surface seawater. EMBL is on land collecting sediment. And then my role is collecting aerosols and microbes associated with those aerosols. So typically the largest source of aerosol you end up seeing in the atmospheric boundary layer is just sea salt. And so those sea salt aerosols are usually produced by wind or wave action. But then associated with that, you can also have terrestrial depositions. So again, wind action sweeping off land can deposit dirt and microbes associated with dirt. And then lastly, you can have bubbles forming. So this is secondary aerosols that produce where you get what are called jet drops, which will then propel small microbes into the air with them. So this is often gases that are produced, can then also propel and lift microbes into the air, where they all basically form the aerosol microbiome. So this is quite exciting for us, because if you were to look at the concentration of terrestrial microbes, you're typically looking at 1 × 10⁻14 per meter squared, and then 1 × 10⁻12 bacteria per meter squared in surface seawater. Part of the challenge with aerosol microbiomes is that we have just up to 10,000 bacterial cells per meter squared, and that's up to, okay? So often we are looking at very low biomass samples, but we're trying to understand how they get from A to B, what kind of genes are they transporting with them, and then also what is it that allows them to survive from one environment to another? So this reduced cell count, it means two things: that you must be a certain size or morphology to be lifted in the air, but then you also need a very specific toolkit to survive in that air too. Effectively, we're looking to explore the diversity of microbes and also their taxonomic and genomic compositions. And we're also looking to understand that exchange from sea to land. And we've been doing so by as I said, we have some super sites which we go to for extended periods of time. This is actually Roscoff Station. I'm unsure if anyone's ever heard of the marine station, but it's quite famous for having one of the largest phytoplankton culture collections. And we were actually fortunate enough to set up our equipment right on this tower here. Our equipment is basically a particle counter. And then although this just looks like a small container you would have your coffee in, it's actually a very powerful vacuum. And so we use that to actually filter for microbes in the air. And we do that for 4 hours for 5 days straight. So working 24 hours a day for 5 days straight, we are changing our filters to try to understand how these things are changing over the time, and to see if we can capture something from land that might end up at sea, or vice versa. So obviously Tara is the other half of our operation. And you can see here, this is that exact same particle counter, which is measuring particles in the air. It's actually measuring particles. I apologize about the sound. That's wind. All the way down from the mast, and it's able to measure the particles. And then it also takes a fraction of those particles into the wet lab area here. So this is underneath. And we're also collecting for elements there. So we can actually do elemental compositions. So we can maybe tell where the particles are coming from. And then above that, so just as I talked about that little canister, we're also sending up that same vacuum all the way to the top of the mast and collecting aerosols there. But then, yeah, so far it has been quite successful. This is some of the particle counts from Tara that I'm showing you here. And one thing you can see is that through Rotterdam, which is one of the largest marine ports, it has been particularly dirty, let's say, we think. But we're very excited to see what microbes are associated with that. And then furthermore, the land team is going to some really diverse locations. So we're very excited to look at the different DNA that's coming from that, particularly the cow DNA. That will be very exciting, okay? But okay, that's about it. I will just thank all of the people that I work with and collaborators. It's always very, very difficult doing things by yourself. So I've been so fortunate to have a great team with me too. So thank you for your patience and attention.